I'm Doug Lowy, Deputy Director of NCI. We really have an illustrious group of panelists, Peter Vogt, Rakesh Jain, and Carlo Croce, Webb Cavity, and Steve Rosenberg. I'm going to throw out 3 questions, and the panelists will go in order to try to respond to one of those 3 questions. But we really want for the participants here, to ask questions, make comments, et cetera. We'd like this to be interactive. So question number 1 is, what do you know now that you think is important that you didn't know recently? Question number 2 is, what do you see as a major opportunity for the near future that we aren't necessarily thinking about? And question number 3 is, what do you see as major challenges going forward? So I don't think that we're going to get comprehensive answers, but it's really important for people to have great ideas. And that's really what we've been hearing about all day today. But it's not just a question of having great ideas, but then putting them to experimental verification. So, Dr. Vogt, why don't you start things off?
Well, I'll start with question 1. What do you know today that you didn't know a few months ago that you think is important? And the simple answer is the involvement of non-coding RNA in the regulation of the cell. Non-coding RNA represents 98% of the human transcriptome. We have ignored this for much too long a time. There have always been voices saying, no, you can't ignore this. You have to start working on this seriously, technically and computationally. And now data appear in the literature that are experimental data that show how important non-coding RNA is. So for instance, in the nucleus, there are RNA condensates that are critically important in controlling transcription. These condensates contain a lot of non-coding RNA, and the non-coding RNA plays a regulatory role. So if we look back, one of the notorious oncogenes, like MYC, is operating only in conjunction with non-coding RNA. I think we are looking at a major sea change in our understanding of the cell, in our understanding of life, because we have to take into account all the activities of non-coding RNAs. And there is a swelling tide of knowledge that will force us in that direction. It's going to be a revolution, probably a slow revolution, because we are all invested in that simple idea of life with DNA on top, then RNA mainly as a carrier of information, and protein. Don't get me wrong. I think proteins will remain standard drug targets for the foreseeable future. But there will be an upswell of RNA work that will redirect our thinking and the focus of cancer research.
Dr. Vogt, thanks so much. Dr. Jain.
So when I started my career, I spoke with Josh Fiedler, the late Dr. Fiedler, and asked his career advice. So he said, well, think about your career as a car. You can either work on the engine or the hubcaps. And at that time when I entered the field, the area of tumor microenvironment was the hubcaps. You could just polish it, but the car is not going to move. But what we heard today from many speakers is that the tumor microenvironment is really not the hubcap, but part of the engine. And so what I learned today from many of the people who I associated earlier, strictly working with the cancer genome now expanded their work into how the microenvironment affects not only the tumor growth, but metastasis and response to therapy. So my thinking is that as the new technologies emerge of not only spatial transcriptomics, but also more sophisticated imaging, we're going to see a better understanding of the microenvironment in real time and how this can be incorporated in cancer treatment. And AI is going to make a major impact in it. It's already making an impact in the detection of cancer. Things that the radiologists could not see earlier can be seen now using AI tools. And there are many laboratories around the world and scientists who are working on it. So what I did not know at the beginning of my career, that area will become so central to cancer research. And I think this is just the beginning, in my opinion. And we're going to begin to exploit the microenvironment more than we have already, which has, as you heard from our very first speaker today, Jim Allison, has already transformed cancer treatment. The word of cancer and cure were never, ever in the same sentence until the immune checkpoint blockers came in. And the survival curves he showed were tremendous. And one area, for example, that has made a difference is that repairing the blood vessels of tumor using anti-angiogenic agents can improve treatment outcome. And already FDA has approved 7 combinations of anti-angiogenic agents with immune checkpoint blockers. And currently, there are about a couple of hundred clinical trials going on this around the world. So we're going to see more of that. So I think that's where I see what I've learned in the recent years, and that's where I see the future.
Thanks, Dr. Jain. Dr. Croce.
I share the opinion of my friend Peter Vogt. What we do not understand enough is the function of non-coding RNA, particularly microRNA. We already know that transcription factor regulate microRNA, and microRNA regulate transcription factor. We already know that alteration in the expression of specific microRNA cause cancer. For example, loss of miR-15 and 16 in CLL and AML, or overexpression of miR-155 in acute lymphoblastic leukemia. And we know from experimental model that dysregulation of a single microRNA can cause cancer. So we can validate this. There is a problem also of tumor resistance of cancer to drugs. You know, if you treat a cancer with one drug, it's quite likely that some resistance will develop. But if you kill a cancer cell with 2 or 3 drugs that target the same genetic alteration of the cancer cell, it's quite likely that all the cancer cells will die without the development of resistance. So I think that the field that is not that explored, is explored but not enough, is the field of non-coding RNA and their role in cancer pathogenesis. And the reason is that to work on this field is not as simple as working in the other areas of cancer research. And therefore, a lot of people are not interested or are not intelligent enough to study this. So I think that that is an area of research that is very important. And you will see in the next few months or few years how important it is.
A prediction. Dr. Cavani.
Well, I'm not intelligent enough to work on microRNA. So I think if we're going to blue sky this, I think what we really need to know, it was hinted by Peter, but I think goes even farther. And that is the structure, the 3-dimensional structure of nuclei, the 3-dimensional structure of subparts of the nuclei, this 3-dimensional structure of how all of these processes work, so that we can understand how these things like microRNA or non-coding RNAs, how they actually exert their regulatory effects. Peter said that there were things called condensates in the nucleus now. This is a topic which is just starting now. And these are sort of non-membrane bound aggregates in the cell nucleus. And I think that's just the beginning. I think that there's more and more of these things. And of course, we're looking in 2 dimensions if we're sequencing, in 3 dimensions maybe if we're modeling. But we need to be looking even more than that, because we need to be looking in the time element too, because these things are changing as time happens. And the question is whether these kinds of 3-dimensional physical structure in the nucleus or in the cytoplasm for that matter, or even between cells for that matter, actually impact on sensitivity or resistance to various therapies. So for example, you could imagine that a therapy that targets a non-coding RNA or involves a non-coding RNA in its mode of action might be quite differently effective in cells which have a crystalline kind of nucleus compared to a more liquid kind of nucleus. And we have no idea about this. Certainly we have no idea in vivo. We're getting some ideas in vitro. And the thing that's maybe most immediately important about that is how the initial diagnoses are made. So we all heard about H&E slides. But really the information in an H&E slide, besides the pathologist's pattern recognition, is what the cells actually do and what they look like in 3 dimensions. We think of cells as round balls that surround other tumor cells, which are round balls, except they have spikes maybe, or they're different colors. But actually those cells, for example, T cells that surround tumors, are not round in many cases at all. They're all spindly and wrapped around cells. So it may be that the shape, which is what I've sort of been talking about, is actually a very important component that we're missing because we're doing the reductionist look down at 2 dimensions as a sequence or as a panel. So that's where I think we're going to go. I don't know exactly how you would even address that, but I'm not smart enough to know that. So I'll turn it over to Steve, who probably is.
Dr. Rosenberg.
Well, we'll turn back to immunotherapy. And what has plagued progress in immunotherapy over many decades was a lack of understanding of the nature of the target that lymphocytes could attack. And what we've learned over the last 10 years or so is finally identification. We finally understand what a cancer antigen is. What does the immune system recognize on a cancer that's different from normal cells? And interestingly, it turns out to be the critical difference between tumors and normal cells, and that is tumors were originated because they developed mutations, and the accumulation of those mutations led to metabolic changes that caused the cancer. Well, when we were using cell therapy to treat cancer patients, we could identify cells we started in melanoma patients that had a response rate when administered to patients a little over 50% by RECIST criteria. We could divide those patients into responders and non-responders, and because we had the cells that we had administered to those patients as a living drug, we could compare the populations that caused regression compared to the population that couldn't. And when we talk about regression in melanoma, as is the case for many of the checkpoints, if you can get a complete regression, and certainly in melanoma when we first did this over 30 years ago, you can get cures of patients out now 25 and 30 years. Well, by comparing patients that responded and didn't respond and studied the test tube full of cells that caused response compared to those that didn't, we learned finally what was being attacked that could cause the complete durable regression of melanoma. And the target turned out to be the proteins produced by the very mutations that were involved in causing the cancer. And so when we looked at, for example, 86 consecutive melanoma patients, we could find over 150 targets that our T cells recognized in patients that appeared to be cured by the cell transfer. And they all turned out to be mutations in the genome. Now, when we began to extend those studies to other diseases like breast cancer, colon cancer, the same things occurred. When we could identify cells that recognized unique mutations in those cancers, in the cancers that those patients bore, we could find the antigens being recognized. And once again, they were the mutations that were involved in the cancer oncogenesis. So we finally understand what cancer antigen is. It's a product of a cancer mutation, and in some sense it's ironic that the very genes that mutated and caused the cancer can, I believe, turn out to be the Achilles heel of cancer approaches that involve immunotherapy. Now, the good news is if cancer mutations are effective targets for immunotherapy, and virtually all cancers have mutations, if we can learn how to target these neoantigens, these proteins, these mutated proteins caused by these mutations, we could potentially develop treatments potentially across all cancers, because all cancers have mutations. That's the good news. The bad news is the exquisite individuality and specificity of the T cells that recognize these neoantigens arising from mutations. In melanoma patients, for example, in 86 consecutive patients, every patient recognized something different. There were no two patients that recognized the exact same mutation as the target of their T cells. And when we then extended that to what's now 205 patients with the common epithelial cancers, it turned out that 70 to 80% of all cancer types, that is starting from ducts and the major epithelial cancers, they too gave rise to T cells that recognize mutations, and that was true for 80% of virtually all cancer histologies. Again, the problem is, with very limited exceptions, in those 205 patients, we could identify over 360 individual antigens. And with the exception of one antigen, which was a KRAS G12D mutation, a particular mutation in an oncogene KRAS, every patient recognized something different. So we have to recognize that treatments with immunotherapy may have to be extraordinarily personalized so that we can develop methods for treating that particular patient, and unfortunately those treatments would not be suitable for other patients. One of the elements of the genius of the checkpoint modulators, as we heard from Jim Allison earlier, is that it can unleash immune reactions that exist. And in melanoma, we have so many mutations that unleashing the T cells that recognize them are sufficient to cause regression. That's not the case for the overwhelming majority of the solid epithelial cancers that still cause 90% of all deaths from cancer. So it's this personalized nature of treatment that we're going to have to increasingly recognize if we're going to make progress in developing immunotherapy. Now, there are some shared mutations. In fact, astonishing how few there are, as this genomic revolution has given us so much information, how few mutations are actually shared. And if you do particular targeted sequencing, you can identify now T cells that recognize KRAS and P53 mutations that collectively KRAS mutations present in about 30% of all cancers, P53 in about half. And so one of the current challenges is to try to identify the T cells that can recognize mutations in those shared molecules, but that's turning out to be a very difficult task. But I think we at least now see our way towards potential solutions to extending immunotherapy to many additional patients.
Thank you. I think that what Dr. Rosenberg is talking about, to me, is the issue of scalability. And I would like to kind of turn that around and say that the technology that we have today, as we have heard, really is extraordinary, where we can try to understand, as some of you have said, you know, whether it's what's happening at the plasma membrane in the cytoplasm, the nuclear envelope in the nucleus, the DNA, RNA, and then at the single cell level, the importance of the microenvironment. How do we take this myriad of information and try to essentially make it practical for dealing with patients so that we either reduce their risk of cancer or we improve their outcome if they develop cancer? This is obviously a big question. We're not going to get all the answers, but I was wondering if any of you might like to tackle how do we go from the incredible amount of information that we have to trying to do something practical for people.
So I think, as you know, the AI is beginning to address some of that, and it's only improving exponentially. So right now, we cannot do this, but the tools are being developed, and I'm sure you've been seeing some publications in New England Journal of Medicine and other places where some of this has already been applied. But I really think that's the tip of the iceberg. I think we're going to see that an exponential growth in that area in the next decade.
Other comments?
Well, scalability is a function of what we understand. When we first developed CAR T cells for the treatment of patients with diffuse large B-cell lymphomas, with B-cell malignancies in general, and pharmaceutical companies would come through and say, if you're creating a new drug for every patient by using their own cells, if we had the disease, they said, we'd come to see you, but we don't see how in a pharmaceutical company we can commercialize such a complex approach. And that happened until one of my former fellows, Ari Beldigman, came by and decided to form a company called Kite Pharma to enable large numbers of people to receive this kind of treatment. And the genius of modern industry developed a technique thereby that one could transfer cells to a central facility, have them converted into the right drug for you individually by inserting the right receptor, and then send it back. And that turned out to turn what seemed to be an unscalable problem into one that was indeed scalable. And now there are hundreds of thousands of patients in this country, in Europe and Asia, even more, that are receiving this therapy that was thought to be non-scalable earlier. And so I think if we can develop T cells against the unique reagents in each individual cancer and figure out ways to find them quickly, and we can find them within about 3 or 4 weeks now, then I think we'll develop scalable approaches that wouldn't seem possible today. Incidentally, when Ari Beldigman started Kite Pharma in 2012, when we transferred our technology to Kite Pharma 5 years later, that company was sold to Gilead Biosciences for $11.9 billion. To me, a proud example of how information that's developed at academic institutions, as well as the governmental institutions like the NIH, can get translated to large numbers of people.
Thanks. If there are comments or questions from you all at the tables, it's very hard. I feel like I'm in an interrogation room, you know, because of these intense lights. But please stand up and say.
Yell. We can't see you.
I would like to ask Dr. Vogt and Dr. Croce, who talked about non-coding RNAs. What about the issue of being able to deliver the inhibitors or whatever to that? One of the principles that we think we understand about cancer treatment is that you need to try to target the vast majority of cancer cells. And how do you deal with that technical issue when it comes to some kind of nucleic acid approach?
This is a very important technical issue because today we cannot target 100% of the cancer cells in a tumor. with a payload of, let's say, microRNA that will do the job. So that can't be done. Another problem is that some of these non-coding RNA, for example, microRNA, have multiple targets. And we know only some of them, okay? And we know that when those some are altered, what would happen, but we don't know the same for all of them. So the possibility of side effects are enormous. And in fact, I know that a few clinical trials that have been done with MIR34 were very bad because it caused illness and death, in fact. So the answer is you cannot do that. But you can do another thing. You know, microRNA have multiple targets. And therefore, when one microRNA is dysregulated, you have the dysregulation of multiple targets. And so from the dysregulation of those multiple targets, you can pick up a good target. I give you an example. ROR1 is an embryonal antigen which is not expressed in any somatic cell, but is expressed in a lot of cancer, okay? So ROR1 is expressed when MIR15 and 16 are lost, like for example in CLL. So in CLL, you have MIR15 and 16 loss that lead to the overexpression of an oncogene called BCL2. And if you use venetoclax, which is a fantastic drug that kill 99.99% of the leukemic cells, okay? But those cells that overexpress BCL2 also overexpress ROR1. So you could treat those leukemia with venetoclax and antibodies against ROR1 or small molecule against ROR1. So the probability that a cancer cell will make it will be very little. And the possibility that resistance will happen will be very little. And using this approach, you could get 3, 4, 5 specific targets that could be used for therapy.
Dr. Volk, did you want to make any comments?
Well, there is a huge technical challenge working with RNA. There are no RNA drugs. But ultimately, what we are talking about and thinking about is the RNA has a regulatory function that is transmitted by an interaction between RNA and protein, RNA and chromatin, RNA and transcription factors. So ultimately, I think that the RNA binding domains on proteins will be an indirect way to get at the RNA itself.
Thank you. Yes.
So as a computational biologist, I've heard a lot of talk about AI coming out that people are sort of hopeful for. And I guess the question I have for the panel is if we're going to use large language models or other tools that are non-interpretable, what are the barriers to translation? And do we need to be sort of focusing on models where we know the underlying mechanism and can do preclinical work? Or if we have a model where we're just going to get a prediction but we don't know what's under the hood, is that going to be effective?
Anyone want to take a stab, respond?
So
Dr. Jain.
we are developing models both ways, from ground up based on mechanisms. And we have developed model for immune therapy and many others based on the understanding the tumor biology. And what we are trying to do is bring things from top down, from large language models, and see if we can merge the two. We have not done that yet. We are currently finishing up a story on microbiome in collaboration with Jen Wargo at MD Anderson, where our team has developed the mechanistic model based on our current understanding, which is very limited, of microbiome's role in cancer response to immune therapy. And their group has developed information using large language models to the data set they have, and we try to merge the two. And hopefully we'll be able to do that in several months or 6 months or so.
Thank you. Others? Jonathan?
So for 50 years, we've spent a lot of time defining the genetic abnormalities of cancers and have identified a lot of oncogenes, and we've developed excellent chemical matter against them in many cases. And yet the arc for those drugs, and I think this will be the same for RAS, unfortunately, is that they work until they don't work, which is rather rapid. And so I think we've heard bits and pieces of why that might be, but I'd be curious what the panel thinks. What is the missing element here? Is it going to be a combination of attacking the tumor driver, if you will, or drivers, plus microenvironment in some fashion, or plus RNA? The drug resistance is such a huge thing after all the investment we've made. Where do we need to go?
Great question.
The question, what I was addressing was the question of resistance. If you have billions of cancer cells in the body of a patient, you will always have some mutant or variant that are resistant to it. So you have to find a way to hit the cancer cell 2 or 3 or 4 times, okay? Now, what gives you that opportunity? The microRNA dysregulation gives you that opportunity. Because if you find that one microRNA is dysregulated and is responsive for the malignancy, you can find them 4, 5, 6 targets, okay? And then you can test those targets, and you can find out whether, in fact, if you use 2 or 3 of them, you have no resistance anymore. I gave you the example of venetoclax and ROR1, and ROR1 antibody or small molecule. In that case, you have 2 proteins that are dysregulated because of genetic alteration in a single microRNA, okay? Now, that is translated in the aberrant expression of 2 proteins. Now, you make drug against those 2 proteins, is quite unlikely that the cancer cell will resist. And by using this approach, you can make target 3 or 4 or 5 targets, okay? Then the cancer cell will have no chance. You will not have any resistance.
But Dr. Crochet, what about the issue if you're using multiple drugs simultaneously of potential toxicity?
Maybe not. You know, for example, with venetoclax, there are very little side effects, if any, okay? Now, you know that ROR1 is only an embryonal antigen. It's not expressed in any somatic cell, so it will not have any effect on any tissue of the body. So why not to use 2 drugs that attack 2 different targets? And one we know already is very safe, venetoclax, and the other probably is very safe because it's not expressed in any normal tissue.
Other, yes.
Thank you. One quick thing I really love when Dr. Rosenberg mentioning that the difference between normal and tumor are the mutations. Because I think maybe there's a misperception that immuno-oncology is non-precision. Actually, incredibly accurate, right, to recognizing 10 to the 18 different epitopes and unleash those T cells. I find that sometimes the world has now again, turned the back at the immuno-oncology, going after ADCs and oncogenic drivers, and forgetting that actually your word beautifully mentioned is a living drug. Right after 10 years of CAR T, the cells are still living there. The medicine is still in patients' blood. So I'd love to see your view of this, the future of oncology, this kind of divergence of half the field are chasing after targeted so-called position, and then the IO is returning to a sort of forgetting land.
Thank you. Does anybody want to comment on that?
Steve.
There was a question behind.
Oh, I have a question too, on top of that. But you want, Dr. Rosenberg wants to.
Did you want to?
Sure. So I was trained as a surgeon. If you have a cancer and it's there, you take it out. It's very simple. The problem, of course, is that once it's spread, surgery, which is still the most effective way to cure metastatic cancer if you just have oligometastatic disease, we don't have good drugs that can eliminate the last cancer cell because of heterogeneity and other factors. But one of the advantages of the immune system, and especially of using T cells in a patient, now that we understand that 80% of all cancer patients have T cells that can recognize their cancer, boils down to we know what the bullet is, the T cell, from that same patient that can recognize the antigen. We know what the target is. We can now begin to approach treatments with immunotherapy without having to understand a lot of the basic metabolic changes and mechanisms, genomic changes that have been discussed. And that, to me, is one of the reasons why I think immunotherapy is going to continue to develop as a major new treatment, because we can, by targeting the antigen we know with the cell that we know has the right T cell receptor, reduces the problem to what are the microenvironmental factors that prevent that T cell from working. And that reduces the problem to a more manageable question. And I think progress is going to be made, and is going to be made rapidly over the next 5 to 10 years.
I have a follow-on question. Really appreciate Dr. Lowy's question about how do you bring this large amount of information and fast advances reduced to something actionable for patients. We learned 85% of patients are treated in the community setting, and how do we motivate the oncologists and doctors, physicians in the community to keep up with this large amount of information and to overcome other financial or lack thereof incentives, as well as in a policy environment to really bring it to bedside?
Big questions.
What you're questioning is, why is there such enormous diversity in the availability of care among different populations? I remember hearing someone talk and said that the best predictor of your outcome from virtually any disease is not your genetic code, it's your zip code.
Yep.
And so one of the major problems, and our new director of the NIH, Dr. Bertagnoli, is an expert in this and is going to make this a priority, is how can we take what we do know and distribute it more equitably? That's the other side of cancer care, and that is not only developing treatments, but being sure that a current system that exists for delivering medical care can supply them to patients. And it's shocking in a country as wealthy as the United States how we do not have a medical care system that equitably distributes effective care among its citizens.
So I'm going to ask the last question, but we have 2.5 minutes, so I'm not sure we will get a complete answer. We've heard a lot about the importance of mutations, and in some ways, Dr. Topalian's description of Merkel cell carcinoma with viral antigens, the classic foreign antigen, and the ability of immunotherapy to do that, to take care of that, or at least ameliorate the situation. But we've learned an enormous amount about signaling. And many proteins where there is signaling are more or less constitutively active, but not mutated. And so my question is, should we start thinking about constitutively active proteins in addition to mutant proteins, perhaps not so much for immunotherapy, but as possible other targets if we think that they are important pathogenetically.
We still have that problem of targeting cancer-specific mutations in some of the most important signaling proteins. A good example is PI3 kinase. We know that there are gain-of-function mutations. There's only a limited number of mutations, 3 mutations, and although there have been claims that there exist mutant-specific inhibitors, so far they haven't been backed up at all. So there is a huge challenge out there for some of these mutations. And PI3 kinase is an example where you have just a single nucleotide substitution. The proteins are very similar to wild type. There are structural changes which we don't completely understand with respect to what's the mechanism for gain of function. But I think there's progress there to be made and progress coming where we can target the mutated protein and leave the wild type, which is important to the cell, leave the wild type intact. We have much fewer side effects.
Thank you. Anyway, I want to thank everyone on the panel. I thank for your comments and questions, and I think we need to stop there because we need to be ready for the next session. Thanks, everyone.